Welcome to the December Soils Network of Knowledge webinar. So I'd like to now introduce Dave Mitchell, who's presenting today's webinar on soil and water, and a lot of you did ask for a webinar that around this topic. So Dr. David Mitchell has over 23 years of experience in soil physical research. He has a PhD in surface hydrology, and he spent a number of years researching components of agricultural water balances. Dave has experience in irrigation, in subsoil constraints in western and northwestern plains of New South Wales, and also was heavily involved in the salinity research when it was going on in DPI, amongst other things. So I'll hand over to you, Dave. I'll just give you presenting rights.
Okay, well, thanks everyone for coming or listening in. It's going to be a reasonably general talk today, just about some concepts, to some overview of some surface hydrology concepts. Really just going to run through some basic understanding of what runoff is, how it occurs, what processes occur, and what some of the theories behind where runoff occurs within different landscapes. I'll have a bit of an overview of some of the soil water concepts. I've just called them soil water concepts, but it's really about what is soil moisture, what is plant available water, and some of the parameters that change or influence the plant or the soil water holding capacity. And then on the last couple of slides are just a quick overview of types of measurements, the typical soil water measurements or techniques that can be used, and some of the issues there you may want to consider when you're setting up or undertaking soil water measurements. And so some of the there is not one size fits all sort of soil water measurement technique. So individual technologies suit individual situations. So we'll just have a quick run through that. But I'll start off with just going through some of the runoff processes. Two types of runoff that can occur. There's rainfall excess runoff and saturation excess runoff. What we've got here in the rainfall excess is, I have to apologise for all these slides. This slide's about 20 years old. It was for my PhD confirmation, and I was working in sugarcane at the time. So what you see on the screen is my attempt of trying to draw some sugarcane rows with some sugarcane hills to explain to the faculty what rainfall excess was. So rainfall excess is when rainfall exceeds the infiltration capacity of the soil and you get excess water ponding on the surface of the soil and then running off. So if I can do the animation, so you get rainfall coming down, you get a build-up or a saturated zone somewhere within the soil, usually towards the top of the soil. Not the concept here is that it's just a really small, really shallow layer that gets that stops infiltration, and then once you get that excess water ponding on top, you get rainfall coming off to the side, runoff occurring. So this rainfall excess is pretty typical occurring in your summer storms when you get high intensity rainfall occurring and the soil just can't infiltrate that amount of water and the excess water builds up and runs off. The other sort is saturation excess runoff. So it's again where your rainfall comes down, but what happens is your whole of your soil profile gets saturated, and so physically no more water can enter the soil, and you get runoff occurring then. And this typically occurs during winter months when you've had lots of little bits, you have low plant demand, so there's very little water being extracted by plants for use, and you're getting the winter rainfall, and it accumulates, saturates your soil, and then at that point any additional runoff, any additional rainfall just usually goes as runoff. So they're the two dominant, or they're the two processes that cause runoff. The rainfall excess is sometimes referred to as Hortonian flow, just named after an American hydrologist who initially described that phenomena back in the late 30s. And saturation excess is always usually called saturation excess. So they're the two types of runoff processes. Now, where runoff occurs in the landscape depends. It depends on lots of different things, through from soil texture, soil structure, topography, slope, aspect, and also antecedent soil moisture. So areas that may be wetter than others tend to run water more than other areas. Your landscape closer to your natural drainage lines tends to be wetter because of the topography, and they tend to run water more than other areas in your landscape. And this is called the variable source concept. So, which means not all of your landscape contributes to runoff. Some areas contribute more to runoff than other areas. Some places in your landscape may never run water, and some areas may run most of the water. And so it's just to bear that in mind when you're thinking about where runoff may occur, that it's quite variable. It varies through time as well as space. So not all areas of your catchment run water, and additionally, as the rainstorm or the rain event proceeds, some areas may become saturated and or exceed infiltration capacity of soil and run water, and so contribute only in parts of your runoff event, while other areas may contribute much more over the whole runoff or storm event. So this variable source concept, and it's just a way of thinking about how runoff occurs. So the next one really is so we tend to measure runoff. Runoff is measured usually just either in the depth in millimeters, or it's normalized, which means we divide through, the amount of runoff is divided through by the rainfall total, and that index is then called a runoff coefficient. And it's just a useful way of looking at how runoff can be compared. And so this is just some long-term data that we've got from a grazing site near Orange, and what you can see, the seasonality of runoff, and you can look at, and probably from the seasonality you can infer the type of runoff that's occurred. So you can see that most of the runoff occurs, if I can get my mouse, in July and August, and that, because I know the site, is all saturation excess runoff. So the site it's in a high rainfall zone, and it gets quite wet during those periods. It's quite cold, so we get low plant demand, and so the rainfall that occurs, you can see if you're reading off on my right-hand scale. So this here, about 20% of the rainfall in August and July, or between 15 and 20%, is runoff. So whatever the totals is for July and August, I can't remember off the top of my head. 20% of that is converted to runoff, and as the temperatures tend to warm up and you get more plant demand, you get a soil water deficit, your runoff decreases. And then you get those summer storms that are pretty typical around, or particularly the tablelands around Orange, and you're getting thunderstorms rolling in December, January, February, and you can get high rainfall occurring, and particularly in single storm events. And these are typically rainfall excess because of the intensity of the storms that occur. And then even just noticing the fact that we don't tend to get much runoff, if any, in April, May, in late autumn, and that's just probably the nature of the climate and coming out of a drier summer. Most of the rain that it does occur tends to occur in smaller showers, and most of it's infiltrated. So it's more about the seasonality of runoff. And that's really all I really want to talk about runoff. I'm quite happy to talk about runoff for quite a long time, but I'll get onto some of the other aspects of surface water balance, surface hydrology. I'll flip to now talking about soil moisture. So soil moisture is just water that's retained by the soil. We tend to be interested in the two limits, we call the two limits to soil moisture, so the wet end, or the wet limit, and also the dry limit. So the wet end is, once the soil is saturated, we usually give it period of time, 24 hours or 48 hours to drain, and whatever water is left over is usually, that's defined as fill capacity. Sometimes called the drained upper limit. They can be interchangeable. There is some slight difference between the two, but the drained upper limit and fill capacity, effectively, once you've saturated soil and you've allowed it to drain under natural conditions for about 48 hours, they're fairly similar aspects. You can measure them in the lab, and you do by applying a pressure in a pressure chamber or a pressure cell, and the pressure is either 0.3 or 0.1 bar, depending on I guess which paper you want to read or which methods you want to employ. 0.3 or 0.1 bar is usually referred to as fill capacity. Now the dry limit is there's a couple of here. They are different, but what they're trying to encapsulate is at what point can plants no longer extract water. And so there's one that's commonly used called the crop lower limit, and that's usually done within the field where you wet up an area and allow a crop or a pasture to grow, and then you put out a rain out shelter over the top and allow the plants to grow without rain for a number of months, particularly in spring when it's actively growing, and then you monitor the soil moisture, and when you've determined that the soil moisture is no longer changing, that's what's determined the crop lower limit. It's slightly different from the permanent wilting point. A permanent wilting point is effectively the same concept, but it's the point when if you wet the soil up again, the plants actually will not respond. And then in the laboratory, again similar to the wet limit, you apply a pressure in a pressure cell, and that pressure is 15 bar. They're all quite similar but different concepts, but they're all trying to give an idea of what level of soil moisture is the lower point at which plants can no longer use effectively soil moisture. So that defines the bucket. It's useful to think of it as a bucket, and when it's really wet, the bucket's nearly full, and when it's really dry, the bucket's nearly empty. Conceptually, it looks a bit like this. So here's a graph of soil water, volumetric soil water on the x-axis and depth on the y. So here are a number of lines out to your right. So the first one is physically the total porosity, so the air spaces within the soil as the colloids and the PEDs are packed together. You can never fill the soil up totally because there is always going to be air spaces that will not fill for various reasons. So just the next one down is when you actually saturate your soil, your larger pores are filled, and then when you allow it to drain, those larger pores tend to drain under gravity, and then you're left with this drained upper limit, which effectively is fill capacity as well. And then you've got this blue shaded area, and that's the amount of soil moisture that's available for plant growth, and that's what people refer to as the bucket. And then you've got this crop lower limit. And what you see is that the drained upper limit and the crop lower limit converge at depth, and they converge not because of soil properties but because of plant properties. Effectively, at some point, their roots cannot physically extract the water from depth. So this is where the crop lower limit differentiates itself from permanent wilting point, or from the 15 bar measurement. If you measure 15 bar for this soil, it may actually continue down here, but physically roots could not access that soil. And this also where subsoil constraints come into play as well. There may be sufficient soil moisture at depth for plants to access, but there may be a chemical or a physical constraint that limits the roots to access that water. So that's the usefulness. This crop lower limit is actually determined by how plants can extract water. So the crop lower limit really is a biological limit, while the drained upper limit is a soil limit. I'll just probably flick along exploring this concept of field capacity or crop upper limit, drained upper limit, and permanent watering point, and the sort of the slight differences, but also the similarities. As you move along the x-axis, you're getting a heavier texture. So the sand is quite a coarse, lighter soil, and your the ability to hold water is not as great as, say, a silt or a loam. You hold more water within those soils, but because it's a coarser texture, it's easier for plants to extract water at lower water contents, because the pores are bigger, and the potential, the soil water potential, or the energy required from the plants to extract that water is lower. The flip side to that is that at some point, because they're very large pores, they don't hold a lot of water, and it drains very readily. And as you go along the soil texture class and get heavier and heavier soil, your pores become smaller, and so the ability to hold more water becomes greater. But then the plants need to use more energy to extract water from those finer capillaries, and so that's why your permanent wilting point increases with soil water content. The idea then is, at some point, your soil texture becomes so fine that plants probably can't extract the water because of the quite fine capillaries, and the energy required to extract that water is not available to them. So that's where it would attenuate right up this end. And a similar thing for field capacity. So the coarser soils, they drain quite readily. So at some point you can't actually, it won't hold water because it's a bit like a sieve. And as the pores become finer and finer through surface tension, and which is the capillarity, it retains water within the matrix, the soil matrix. And so that's where, at some point, the holding capacity of the soil is attenuated by the actual porosity. So up this end of your field capacity, the actual porosity, the total porosity of the soils is such that it can't take any more water because it's completely filled with water, and the other 60 to 65% is actually made up of the soil solids themselves. One of the other ways of exploring soil water is looking at the drained upper limits and the crop lower limits, and this is just from some of the grey verticiles in northern western New South Wales, and just exploring some of those concepts around subsoil constraints. So, excuse me, I'll just go through this slide because it is quite a complicated or busy slide. The acronyms, lower limit, so you see the wheat LL, it just refers to the lower limit. SSC, subsoil constraints, and on the graph itself, you can see the black line, well, that's your upper limit, your drained upper limit, so that's the wettest the soil can get, and these scanning curves back are the different crop lower limits as they are affected by subsoil constraints. So what we were talking about before, you may, on a similar soil, may measure a whole range of soil water at depth, and you'd expect sufficient soil water or volumetric water content was sufficient to support a crop, but there may be other physical or chemical constraints limiting routes to exploring or extracting that water, and this is what this slide really tries to display. So here's this red line, and that's the typical extraction patterns for wheat. So you can see, if you look at the area between the red and the black line, you can see how it's quite a big volume there, and it's quite a lot of water, and so your bucket's quite big. And as you get subsoil constraints present in your soil, you're constraining that the roots so they actually cannot explore that soil depth and extract water. And as your subsoil constraints become more and more present, the bucket size becomes smaller as the roots are prohibited or limited to exploring that soil depth. So the plant available water for, say, this blue line this soil here, even though they're very similar soils, is quite different from this purplish line here, and it's because of the presence of chemical or physical subsoil constraints. Now I'll just move to a bit more about plant available water. So what we tend to do is we'll sum up the volume of water between the two lines, between the lower limits and the upper limit, and we usually call that and then sum that up over a depth of interest. And so for this one, say the blue line, it's around no water is extracted below about 1 metre. So whatever that area in that curve there, it's summed up as and you get a term as a millimetre. We've done a similar thing in some grazing trials around Orange, and so this term PAWC, plant available water content, is looking at the soil over time, and we've determined from looking at the soil water measurements that no water was being really extracted from below about 800 millimetres from these soils, and that was pretty consistent across the whole landscape. So we then we measured soil water, volumetric soil water to 1.5 metres, but then constrained it to 800 mil, and then worked out what plant available water content was available for plant growth, and then just graphed that out. So that's from basically March 2008 to the mid-2012. And we had a couple of different grazing treatments. We were looking at what was the effect, if any, of cell grazing or time-controlled grazing had on soil water. So we had same-size paddocks, and they were split into the one paddock. The one size was either divided into 20 paddocks, 4 paddocks, or 1 paddock. And then we monitored soil water under those different treatments, and you can see that there was very little effect on plant available water content under this grazing. And what you'll notice a few things. Firstly is, as you go into winter, your plant available water content accumulates. We're uniform rainfall down here in Orange, and our winters are quite wet and quite cold. So we get accumulation of soil water into winter, usually starting to accumulate in June, July. By late August, September, we're usually quite wet. And if you think back to that runoff slide, that's when we're getting all that rainfall, all that runoff, particularly in August. So we tend to get very wet, saturated soils in August. Come to September, October, it's warming up and plants are starting to actively grow, and they start to actively extract water out of the soil profile, and you get these patterns. We do get summer storms, so you do get sometimes you get a bounce in soil water, and you get particularly one here in 2010 when it was quite a wet year, and also we got a bounce in 2012. We had a very wet February, March in 2012 when I think there was a few inland floods as well, when Wagga and Griffith got flooded, and we copped it a bit as well. But the thing to notice here is the accumulation through winter and the extraction through spring and summer, and the usefulness of having this sort of information available to make some decisions. So if you're coming into spring, mid-spring, and you know you've got 80 mil of soil water available, you can make an estimate of how much water plants can use, and the estimate that we tend to use is around 2 to 3 mils a day for pasture. So you can start thinking, well, I know I've got 3 or 4 weeks of good growth ahead of me, and then how would I change my stocking or my management of that pasture and or my stocking rates to adjust to accommodate for that growth. So that's what we're exploring in this sort of these sorts of data. The landscape that we did this measurement on was highly variable. So the next slide is effectively the same data, but we divided the landscape up into high, low, and medium production zones. So the high zones were effectively the valley bottom floors, and the low zones were the rocky ridges. And we had about 100 metres relief between the ridges and the valleys. And then we measured, instead of looking at treatments, we just looked at plant available water under these different landscape zones. And you can see what's happening is the valleys, because they're deeper soils, they're better soils, they hold more water, they accumulate more water, and so allow for plant growth to occur for longer into the spring, and if we get lucky enough to get a winter and autumn break, also into autumn. And so what you can see is these patterns where more water use out of these landscape zones. And again, thinking about how you might want to use that sort of information. Again, if you know that your high production zones have 100 mils of water, you may want to manage them quite differently from your low production zones, your rocky ridges, in terms of improving or getting production out of them. The circles just indicate where there's significant differences between the different production zones in terms of soil water or plant available water. The other term you come across is, it's an irrigation term, and it's readily available water. The concept really is, at some point, water is not available to plants, and then there's a range where there is water available, but plants need to put in a fair bit of energy to extract that water. And then there's that area where the water is readily available and plants really don't have to work too hard to get it, and that's this concept of readily available water. I stole this image out of a prime fact that Jeremy Giddings had authored, so I just want to acknowledge that. I haven't acknowledged it on my slide. There are two different languages when we talk, it can be two different languages when we're talking about soil water. One's an irrigation language and one's a dryland language. The readily available water really is an irrigation term, and it's really about when you're irrigating, you really want to operate within the readily available water area, so between field capacity and at some point, this refill point, where plants tend not to have to work too hard or to get too stressed to access soil water. And again, readily available water points, that refill point, is really dictated to by texture. And just the next slide, just again out of that prime fact that Jeremy wrote, looking at just the different, how you define that readily available water and how it's related to the different soil textures. So again, it's again, as you increase in texture, you get finer and finer texture, your refill point becomes lower in terms of the suction plants need to exert to extract that soil water. Soil water can be envisioned or conceptualized as a bucket, and and as you go between the wet end of that, as the bucket falls and it dries out, that's really the best way of conceptualizing soil water. Now, how do we measure it? Well, there's a number of different ways of measuring soil water, and it all depends on why you want to measure the soil water, and really the budget in which you want to do. So the three ways I've noted here is the sensing, where you're not actually measuring soil water, but you're using some way of sensing a parameter in the soil, which then you infer or you've calibrated to soil water. And they're the more proximal ones, or the ones that you usually have to either have in contact with the soil. And the neutron probe, which is where you actually carry around a neutron probe and you have access points and access tubes, and you go down and you measure at individual depths the soil water. And then you have the other types, which are called in situ ones, so you actually install them at a particular site, and you can either usually log them or poll them whenever you're interested in knowing what the soil water is. And they're capacitance probes, which work on the dielectric properties of soil, so it infers soil water, volumetric soil water from some of the soil properties. Time domain reflectometry, which is just another, puts a little signal through the soil and measures the reflectance. And a tensiometer, or versions of tensiometers, which is a ceramic cup or some sort of porous material that measures the matrix potential. And then you can, from that and understanding the matrix potential, you can then estimate soil water content. There are remote ways of sensing soil moisture. The major one is this AMS-3 satellite, which is a NASA satellite, I think, and it uses microwaves. That's the M in the AMSRI. It really measures the near surface soil, and that's quite used. That's particularly for weather forecasting and flood forecasting, because they have the soil properties that drive a lot of the global circulation models or the weather forecasting models, and they need just that near surface soils. I've put in NDVI as a way of measuring soil water. So NDVI is sort of the normalized differential difference vegetative index, and it really just looks at how vigorous plants are growing. And so the more vigorous the plants are growing, then you could probably infer back from then how well they're accessing soil water. They may not be growing vigorously for a number of other reasons, so it's not a direct measure of soil water, so that's why I'm sort of a little bit circumspect about saying it's a measure of soil water. But you can infer, if you had a reasonably good idea that the soil water was not limiting, was not limiting, then you can infer some sort of soil water measurement from that. The two ways of directly measuring soil moisture is actually going out there and coring it, going out and actually extracting a sample of soil, and then measuring the amount of soil water that's that once you dry it in an oven and you measure the amount of water that was in that. Push probes are another way. It's a measure because it's a measure of the amount of water that's as you push a 5/8 rod into the soil, you can get an estimate of how deep that wetting front is. And then the third way is again, it's not a direct way of measuring soil water, but it's a scheduling. So once you worked out your starting point, you can estimate, if you measure evapotranspiration, which is the amount of water that's transpired by plants, you then work out what it's like a bank account. You say, well, if they've transpired 5 mils this day, or it's been 50 mil of transpiration over a week, and then you know how much water you started with, and you've got an idea of how much water's left in your soil, and it's then you can need to apply an irrigation to refill that amount or replenish the soil water. So there are the three sorts, and I guess the message is that they all have their place, and they all are useful ways of measuring soil water. One technique over another is probably, it's really about the personal situation or the individual situation you're requiring to measure the soil water for. It will determine which method you use. And then just some photos. So there's a push coring with a picture of Jane Jenkins from Trangie when we were doing some sampling out at from Nyngan. A neutron probe, which is pretty standard equipment. This is some of the products that get out of the AMSRI. It needs to be, I think, turned into these sorts of things, but this is some of the product that can be obtained from some of the satellite, and these are just near surface soil moisture maps of Australia. And we've got just down the bottom, these are the theta probes, just TDRs, which you install into a soil profile. And an evaporation pan, you can estimate your evaporation or your ET and then schedule. And then this one over here is a capacitance probe, again, just a sensor that you install in your soil. So just to wrap it up, we'll start from the top. So I'm just talking about the two types of runoff, the rainfall excess and the saturation excess. Types of runoff and when they occur. So saturation excess tend to occurs in your winter time when your saturated soils, and your rainfall excess tends to occur with your summer storms or with high intensity rainfall. And then the concept of the variable source, which is just parts of your landscape contribute differently to your runoff hydrograph. There's a range of different reasons why that may occur. And then we just want to just refresh the concept of plant available soil water, plant available water concept. So you've got your upper limit and your lower limit, and those are influenced by a whole range of different factors, as we've discussed from soil texture through to chemical and physical soil properties. And there are many ways of measuring soil water, and you really just have to figure out what's most appropriate for the individual situation. And that's it. So thanks for listening, and I'm happy to take questions.
Great, thanks, Dave. We do have a question here from Peter Smith.
I had two questions there. Just being picky and more for clarity for myself, Dave.
Yeah.
In your graph with the grazing plots.
Yes.
You referred. You called one of the blue lines runoff coefficients. But by the way you had it, yep, that one. It looked more like it's just really runoff percentage.
Well, runoff coefficients, they're a percentage of rainfall.
Yep. All right. Now that clarifies.
So yeah. So sometimes your runoff coefficients can be presented as a percent, or sometimes as a decimal. So you know that should be 0.25 or something. So typical runoff coefficients for the landscape around Orange is usually around 0.04 or about 4%. Well, that's what we thought. That's what some of the estimates were. If you look at the old Cellcon manuals, I've just chosen to present them as percentages. But yeah, percentages of the rainfall, of monthly rainfall.
Yep. So you've summarised actual measurements to derive those percentages.
Yeah, there's a number of different ways of determining runoff coefficients. I've chosen to display them here as monthly totals, while it's probably more accurate to do it as I tend to do it as an event. So you can determine a runoff event. So a rainfall event is, you might get, say, 40 mils in a rainfall event, and then your runoff may occur then for the next 2 or 3 days afterwards. And you can then say, well, of that rainfall, for that 40 mils, 10 mils ran off. Our runoff coefficient is then 0.25. We can do it. You can do it that way, or in terms of just presenting it, it's just sometimes easier to do it as monthly coefficients, because it's just trying to get that concept across that the seasonality of runoff coefficients changes. You can sometimes get annual runoff coefficients, and I can't remember the annual runoff coefficient for this site here. I think it was actually around 8%. So and then there's a whole seminar in talking about runoff coefficients, but I won't go there.
No, mate, that's cleared it up a little bit for me. And second question I have, which is just probably another one of these mere academic points. The minus 15 bar, we're just saying that for all crops as our crop lower limit.
Mm-hmm.
Will we be having any variability in that? Like, what is it? It's sunflowers or something?
Yeah, it was done by a Russian guy. I can't think of the name of the scientist, but it was many, many years ago. And he grew sunflowers in a pot and then effectively dried down the pots till he could not revive the sunflowers, and then measured the soil water and then determined what pressure it was, or the suction it was, and then he came up with 15 bar. And so it was set like that. What we're tending to do more and more, and it's happened over the last, say, 15 or 20 years, is doing more of these sorts of determinations using the crop lower limit, because it's about what occurs in the field that you're really interested in, rather than some sort of artificial or line in the sand that's been agreed on as your permanent wielding point. These types of determinations, while much more harder to do because it's an in-field thing, are much more accurate.
Okay, thank you.
No worries.
Thanks, Peter. I'm sure you're clarifying it up for other people as well. So thanks for your questions. We've got a question from Michael Rose. So Michael's wondering if Dave, if you can comment whether you know of any literature on the impacts of soil carbon on plant available water content? There seems to be a lot of talk about the value of carbon, but not much hard data. Great question, Michael.
Yeah, good question. Okay, it takes me back because when I was finishing my undergrad degree at Sydney Uni, my fourth year project was adding a lot of sawdust to soil to improve the soil carbon content. This was, we were all interested in soil structure back there, and we knew that soil carbon had some role in soil structure. And we tried to measure if there was any change to the water holding capacity of soil just based on by adding a crap load of sawdust. We found there was lots of other issues that we weren't really expecting, and the quick answer would be there's probably no, there really isn't, because the amount of soil change in the soil structure that soil carbon would induce is quite probably quite small. So what you're hoping for is the soil organic matter will increase soil carbon, and that soil organic matter then will improve the soil structure, and then improve soil structure, you'll get a better water holding capacity. But I just think that the levels of, to get any measurable increase in plant available water content by doing that would be quite quite variable, and also you would have to increase it by a reasonable amount. That you could probably do it within small trials, but to do it sort of broad acre, you may not be able to. Just trying to think of the literature. Even some of the work that Dave Mychalk had done in the previous, say, 10 or 15 years ago, using biosolids around the Goulburn area. I'm sure that Dave Mychalk has published that work. I'm not sure where, and I'm not sure what type of measurements they did, but anecdotally I know he's told me that they did get a measurable change in the in water holding capacity. I don't know how they measured it or what techniques they used, but that would be one of the only ones that I could think of off the top of my head. And it's a good idea, but about increasing plant available water content, increasing soil carbon. But I just think it's a very long process to increase soil carbon in your soil, and then any sort of secondary effects may take some time as well to manifest.
Thanks, Dave, and thanks very much, Mick, for a great question. I hear that one all the time. It's really good to look for the hard data on it. You've got another couple of questions here I'm going to read, Dave. So Dave Cordina has written in, and he's asking, available water is expressed in terms of soil capacities and limitations, and often noted for a particular crop or plant. Do different plants have different capabilities other than root depth in mining water and at different pressure? That is, does the crop lower limit and other measures, the size of the bucket if you like, vary depending on the plant type?
Yes, would be the quick answer, yes. So if you can compare something like, trying to say lucerne to, which is a very deep rooted plant, to, I'm just trying to think of a shallow, say subclover, even though they're very different plants. One's a winter active and one's a summer active plant. But again, the differences are the crop lower limit would be quite different for them because the ability of those roots to explore and extract water.
Thanks very much, Dave. Both Daves. So Ed Joshua asks, do pastoralists measure soil moisture? And is there a database of soil moisture monitoring data?
Well, it's something that we are actively exploring, and it's something that one of the projects we're very keen on to look at is trying to get graziers to actually do some of the work, to take on some of this concept of measuring soil water in these, particularly when your seasons are turning, spring and autumn, and then basing some management decisions around your estimates of plant growth based on your soil water. That's an area, and Sean's very keen on that. Sean Murphy up in Tamworth. There are services that are on the web that measure soil water, and there are a number of different networks. I can't think of how, I don't know if any people are actually using them for grazing decisions, but there are a number of particularly academic soil water networks and also land care type networks that display soil water or representative landscapes. So the one that I'm thinking of is AusNet, which covers bits of Wagga, bits of Yanco and parts of Victoria, and that's run out of University of Melbourne. And I know of there are a number of land care groups that also have put in soil water monitoring networks.
Great. Dave, thanks very much for your talk. So thanks everybody for attending.